nearly there. We're just getting the microphone. Check 1, 2. Put that on your belt. Right. Pocket.
This is a nice little feature here.
That helps. Perfect.
Makes a difference.
Maybe we'll give it a couple more minutes.
Sure.
Do you have videos or anything? Videos, audios?
No.
Yeah.
Yeah. Keep it easy.
Yeah.
Yeah. Thanks for the invitation.
Of course. Yeah, thank you for coming.
Yeah, of course.
It's a long way.
It's a long way, but
Sometimes to intimidate people.
yeah.
Yeah.
Yeah, but worth it.
Yeah.
Yeah.
Are you visiting a couple places in this trip or are you just here?
I went to Chicago on, when was that? Thursday? Friday?
Oh, that's great. Yeah.
Yeah.
Is the University of Chicago? Or
No,
is it
UIC.
USC.
Yeah, it's still pretty good.
Yeah. USC is good. Yeah. There were a couple of warm people there, right?
Yeah.
Jeanette
Met with Dan Shea.
and Shea.
Yeah.
There's an electrofeed
Oh.
yeah, recording from neuromuscular junction.
Didn't know that. Oh, Janet Richmond.
Yeah.
Yeah. Yeah, she wasn't there, unfortunately.
Yeah.
And I met up with Pascalis. Correct. Yeah.
To introduce Roger Pocket. And Roger's visiting us from Monash University in Australia. Roger started to use C. elegans to study neurodevelopment function during his PhD work. And his thesis focuses on a family of homeobox transcription factors that patterns nervous system during embryonic development. His work moved on to the functional side during his postdoc training with Oliver Hobart at Columbia University. And one of the very impressive studies, Roger showed that the one environmental cue, which is hypoxia, interacted with a neuropeptide pathway to reconfigure a functional circuit that results in animal to display a behavior response that they don't usually display under normal conditions. And then following his postdoc training, he went to Denmark to start his own lab, where they continue to investigate how the internal factors and the external cues interact to regulate neurodevelopment and function. They did a series of beautiful studies. And so one I really would like to mention is that they show that when conserved microRNA surprisingly regulate sugar chain homeostasis, which apparently is very important for the neural migration. So this is one of the pioneer work that characterized how the microRNAs regulate development of nervous system. Right. So today, Roger gonna share with us a couple of unpublished stories from his lab. I look forward to his talk.
Okay, can you all hear me okay? Great. Thanks very much, Yun, for the introduction, also for inviting me to come from Australia. It's really great opportunity to come here and to meet everyone during the day today. So yes, I'm going to talk to you about two completely unpublished stories, actually, today that are coming from my lab, hopefully this year. But first of all, I just want to give you an idea of where I live right now. I've moved to the southern hemisphere, and I live in the city of Melbourne in the south coast of Australia, a really beautiful place to live and work. And my lab is housed in the Monash Biomedicine Discovery Institute, which is formed by these 6 different themes that focus predominantly on development and disease processes. And this institute is really amazing. It's underpinned by fantastic platform technologies and computational biology. And the idea is to have everything in-house, make interesting discoveries in biology, and hopefully try and translate into the clinic. Obviously, that's not always possible, but wherever it is, we really try to do that. So I've spent my whole career working on this critter here, C. elegans, and what I'm showing you here is the entire nervous system labeled with green fluorescent protein. And I suppose the overarching theme of my lab is to try and decipher mechanisms that control how the nervous system develops and also how it functions. There are obviously very important advantages that the worm has as a model, and we try and address a number of different questions in the worm's nervous system. You know, how do you make a neuron? How do you generate particular fates within the nervous system, all the way through to migration, axon guidance, but also more and more so now we're trying to focus on how neurons and circuits function, and also how neurons survive. And throughout my work, we performed lots of forward and reverse genetic screens and identified a number of transcriptional and post-transcriptional mechanisms, two of which I'll highlight today. So I thought, just to give you a flavor of the different kinds of research we perform in the lab, I'd talk about two completely unrelated stories. So what we'll probably do is stop halfway through and have questions on the first story before we move on to the second. So the first story was really pioneered by really excellent PhD student Vaida Diozasute, and she's left me now but published the initial work in this story. But really it's been spearheaded recently by a really amazing New Zealand postdoctoral scientist who worked on Drosophila initially in her PhD, and they made the very sensible jump into worms for her postdoc. So the whole area of this research is to try and understand how the nervous system controls fat levels in the intestine at a distance, and also how the intestine controls behavior through the release of satiety signals. And this mechanism here is required, obviously, to maintain a correct level of fat within the animal, but also we found that it controls whether animals' behavior changes between active, sedentary, and quiescent states. So that's the first story I'll talk about. And the second story is completely different. This is almost 10 years of work, unfortunately, but we've got there now. This has predominantly been performed by two Danish students and postdocs, Camilla and Patrick, where we've identified a new mechanism through which a microRNA controls autophagy to regulate polyglutamine aggregation. Okay, so the first story starts with a transcription factor, and it's an ETS domain transcription factor called ETS5. In mammals, it's called PET1, and ETS5 is a direct homologue of this protein, and in many studies, PET1 has been shown to be involved in the specification of serotonergic neurons predominantly, and as such is important for the regulation of anxiety and aggressive behavior, circadian rhythm, and also obesity. So this is the very first story of my lab, and unfortunately two other labs had the same discovery at the same time, but we all managed to get a publication out, and we first identified that ETS5 is involved in specifying a particular neuron in the worm that senses oxygen and carbon dioxide. So that was the first discovery. I'm not going to talk about that because that's published. During this work, we were analyzing the expression pattern of ETS5, and we found that it's expressed in around 16 neurons in the head, suggesting that it has other functions. So what we did here was to try and identify what other behaviors ETS5 may be regulating. So we performed a behavioral screen, an array of different behaviors that I'm not going to go into. I'm just going to focus on the one particular behavior where we found a very robust phenotype. So this is called expiration behavior, and it's a very simple assay for undergraduates to perform actually, where you place a single worm on a fully coated agar plate. It's coated with bacteria. We let the worm move around for 16 hours. We remove it the next morning, place a grid over it, and ask how many squares did it cross during the overnight period. Now, during this 16-hour period, the worm is switching spontaneously between particular behaviors. Around 20% of the time, the worm is moving forward in a quick manner. around 75% of the time it's dwelling here, where it's exploiting the environment and grazing the bacteria, and around 5% of the time they're absolutely inactive, so they're not moving and they're not feeding whatsoever. It's been shown that the spontaneous switching between these behaviors is influenced by food quality, such that when there's a enhanced food quality, there's increased dwelling and quiescent behavior. Previous work from a variety of labs have shown that roaming behavior is predominantly promoted by a pigment dispersing factor peptide. Dwelling is promoted by serotonin, and quiescence is promoted by satiety. So what's the role of ETS5 in this behavior? Well, it's quite dramatic. So if you look at, on the left here, a wild-type plate, where one animal has been placed overnight, and we can see all these tracks in the bacteria. This is an ETS5 mutant animal that really hasn't explored its environment very much. These worms are not uncoordinated. They've chosen, for whatever reason, to remain in a particular area of the plate. We score this, so there's 86 squares that worms can cross, and wild-type animals cross around 60 squares, and ETS5 mutants have a dramatic reduction here. When looking at the specific behaviors, we found that an ETS5 mutant has reduced roaming and increased quiescence. As quiescence is caused by satiety, we hypothesized that ETS5 may be regulating satiety in C. elegans. And the way we tested that was to analyze the fat levels of an ETS5 mutant compared to wild type. And we've performed that by very simple staining with this molecule called oil red O, which stains intestinal lipid droplets within the worm. And we found that ETS5 mutants animals have an increased fat levels, around 20 to 30% increase in fat levels. So that suggested to us that increased fat levels in the worm, inhibits exploratory capacity in the ETS5 mutant animal. So we performed lots of experiments to confirm that. One simple experiment here that I'll show you. There's an enzyme in the worm called POD2, which when you knock it down by RNAi, specifically reduces fat levels in intestinal cells. So we placed ETS5 mutant animals here, which have reduced exploration, on either control RNAi bacteria or on bacteria where we knock down this enzyme, which reduces fat levels. And we could see that we could reverse this behavior back to wild type. Okay? So specifically reducing fat levels reverse the ETS5 mutant phenotype. We performed a number of rescue experiments, and we could show that we could specifically rescue ETS5 mutant exploration phenotype using its own promoter driving its cDNA. When looking at where this promoter drove expression, we found that it was expressed in two different sensory neurons called the BAG and ASG sensory neurons in the head of the worm, suggesting that it's acting from either or both of these neurons. So we now have a mechanism through which ETS5 regulates expression of genes within particular neurons in the head of the worm and regulates fat storage, which then controls the activity behavior of C. elegans. So that's all the published work. Now we're going to move on to unpublished work for the rest of the talk. So this is where Ava Handley took on the project. We know that the intestine of C. elegans is not innervated by neurons. So that suggested that there's some neuropeptide regulated mechanism at play here. To confirm that, we showed that when you knock down the expression of this gene called UNC31, specifically in the BAG neurons, we reduce expiration behavior. Now, UNC31 is required for release of neuropeptides, suggesting that indeed neuropeptide regulation of this behavior from the BAGs is occurring. So this is exactly where Ava started her postdoc. So we looked at the literature to identify what neuropeptides are specifically expressed or expressed at least in the BAG neurons, and then screened to see whether any of the mutants for these neuropeptides had any expiration phenotype. And we found that mutants for the INS1, insulin, has reduced exploratory capacity. Here are the squares entered in wild type, the ETS5 mutant, and we have a reduction in an INS1 mutant. Not as dramatic as the ETS5 mutant, but indeed there is a phenotype there. We're able to rescue this by re-expressing genomic DNA. So we wanted to ask whether potentially INS1 may be a target of ETS5 in this pathway. First of all, we wanted to check whether INS1 mutants exhibit excessive fat. And I've recently had a Chinese professor that's come to visit for a year to learn all things worms. So Chuli Wu has been doing these experiments for us. We have the intestinal fat levels here shown with oil red O staining. And we show with two independent mutants for INS1, they do indeed have increased fat levels similar to the ETS5 mutant. And again, we were able to rescue this phenotype by expressing genomic INS1. When looking at the expression pattern of INS1, we see that it's expressed in a number of neurons in the head, which include the BAG neurons. This is the expression pattern of the INS1 gene driven by the, or GFP, sorry, driven by the INS1 promoter, and we can see that we overlap expression with the BAG neurons there. And indeed, this same promoter is again able to rescue the INS1 mutant phenotype, suggesting at least that BAG expression of INS1 is responsible for controlling expiration. So now we have this transcription factor and this insulin that both are expressed in the BAG neurons. We wanted to confirm whether this transcription factor is regulating expression. And to do that, we looked at INS1 expression in wild type and in ETS5 mutant. And you can see very clearly here that expression of INS1 is dramatically reduced when you remove the transcription factor, as shown in this graph here. We obviously wanted to confirm that this expression, regulation of expression is direct. And we did lots of experiments here, but I don't want to go through all the details. I'll just show you one of them. We analyzed the promoter of INS1, and we found two canonical PET1 or ETS binding sites around 1,000 base pairs upstream. So we interrogated those binding sites by mutating two important residues here, and then expressed the full promoter with and without the mutation driving GFP. And we saw exactly the same phenotype as the ETS5 mutant, where we see mutation of these sites reduces expression of INS1 in the BAG neurons. And it seems that either single or double mutations have exactly the same effect. So we've now shown that ETS5 is regulating the expression of INS1 in the BAG neurons. We wanted to confirm whether we could directly express INS1 in these neurons to control both expiration and fat storage phenotypes to confirm our findings. So we used a particular promoter called FLIP17 here that's specific for the BAG neurons to drive INS1 expression. And we could see full rescue of the INS1 mutant phenotype, showing that that is sufficient to drive INS1 rescuing capacity for this phenotype. When we looked at the fat staining, we saw the potent effect of INS1. So we have fat staining in wild type, the INS1 mutant here, and expression of INS1 either with its own promoter or a FLIP17 promoter dramatically reduces the levels of fat here in these animals. And these are very low expressing lines. If we overexpress INS1 from the BAG neurons at high levels, worms arrest. And they arrest at the L4 stage just prior to adulthood, and suggesting that there's insufficient fat levels in these animals to warrant production of progeny. And we're investigating that a bit more right now. So we have this regulatory mechanism where ETS5 is directly controlling the expression of INS1 from the BAG neurons, which is released and somehow controls fat storage. It seems this is a conserved mechanism. So in mice, the ETS5 homologue PET1 controls insulin expression in the pancreas. However, its role in controlling brain insulin expression has not been shown. But at least the regulatory relationship is present in mice. So we've shown that this mechanism is required for fat storage. We hypothesized that would possibly need to modify this regulatory relationship in different conditions, suggesting that there may be feedback from the intestinal fat storage back to control this transcriptional regulation. So to dissect that a little bit, we used a trick where we fed worms with a very rich diet. So if you feed the bacteria that worms presented with glucose, they're very, very rich. And worms that eat this are very happy and they store a huge amount of fat, as shown here. So we used this to ask whether changing its environment and fat levels could feed back onto the transcriptional regulation by ETS5. We did this in two different ways. We made a GFP tag CRISPR line. So we have endogenous expression of, ETS5 in the BAG neurons. And we could see when you increase fat levels, we have a reduction of ETS5 expression. And we showed the same effect with INS1. We have a reduction in expression of INS1. So what does this all tell us? Well, in poor food conditions, which is actually what we normally keep worms in the lab, it's a kind of a poor diet for them, ETS5 positively regulates INS1 expression, breaks down intestinal fat, and causes animals to explore their environment to try and identify more optimal feeding conditions. When conditions are good, we have rich food supply, ETS5 expression is reduced, which reduces INS1, and you store fat and exploit the environment that you're in. So what we're trying to do now is try and understand how INS1 communicates to the intestine, and this is extremely complicated, unfortunately. There's a single DAF2 insulin-like growth factor receptor in worms, but there are 40 insulins, each of which can act as an agonist or antagonist, possibly, on DAF2 in particular neuronal contexts. INS1 itself has been shown to be an agonist and antagonist in different neurons. But we're trying to address this through the level of DAF2 at present. Haven't got so far with this, but I'll show you what we have. We obviously want to identify where DAF2 is acting to control exploration behavior, and we actually hypothesized INS1 is released from the BAG neurons, controls DAF2 in the intestine. That's not the case. It's more complicated than that. This is the DAF2 mutant phenotype with regards to exploration. It's a poor explorer, just as an ETS5 mutant. We can rescue that phenotype by expressing DAF2 in all neurons, but not from the intestine, okay? So where we are now is that INS1 is released from the BAG neurons and controls DAF2 from one or multiple neurons, and we're trying to address that at present to control intestinal fat storage and explore the exploration-exploitation decision. So I think I'll stop there. That's the end of that story. If you'd like to take questions now or at the end, what do you want to do?
It's up to you.
We'll keep going. If you want to keep going? All right, we'll keep going. Okay, so now we're going to move on to something couldn't be more different, but hopefully you'll enjoy it. So this is a project that was taken up by Camilla Nehammer, who was a PhD student in the early days of the lab. And it's been worked on by Patrick Eilerskov, who is a postdoc in David Rubenstein's lab at the University of Cambridge, who's really pushed this project forward in ways that I didn't think were possible. So, in the early days of my lab, I was really interested in understanding how microRNAs control development and stress responses. So microRNAs are transcribed into a hairpin structure here, processed into a short RNA, which then targets through imperfect complementarity to the 3' UTR of target genes. And in most cases inhibits translation. This is a highly simplified schematic, but I think sufficient for our needs today. So in C. elegans, microRNAs were discovered 25 years ago. And the first maybe 15 years of the field, very few functions for microRNAs were identified, and even now there are very few functions known. But in the mid-2000s, Bob Horvitz decided, okay, we need to address this, so he wanted to ask, what are the 300 plus microRNAs in C. elegans doing? So he initiated a project to make genetic mutants for as many of these as possible, which is a Herculean task at that time, pre-CRISPR, but he made many mutants, and they said, let's just find phenotypes, no problem. Then he published these two papers. The first paper, most C. elegans microRNAs are individually not essential for development or viability. So they found very few phenotypes for these individual microRNA mutants. He said, okay, so microRNAs appear in families. Let's remove whole families of microRNAs, and then we'll find phenotypes. Then he published this paper. Many families of microRNAs are not essential for development or viability. Okay, so not going so well. So this is pretty much when my lab opened, when the second paper came out. So I thought, okay, we need to address this. So we came up with two hypotheses. One, microRNAs function in specific cellular contexts, and we showed, as Yun mentioned, a particular microRNA that controls migration of a specific neuron. So it's a very particular context. In addition, we hypothesized that certain microRNAs may function in certain conditions, stress or environmental conditions, and we published a couple of papers on that, and we've recently submitted this, this new story. Okay, so this is in the realm of protein aggregation, toxic protein aggregation disorders, including Huntington's, Parkinson's, and Alzheimer's. And worms have been quite a good model for studying mechanisms that control aggregation of these toxic proteins. And there's obviously a lot of controversy in the field as to what is actually the toxic component, whether it's an aggregate or oligomer or whatever, but these are good models to study the basic biology of how these aggregates form. So we asked whether any microRNAs may function in toxic protein aggregation. And there was a paper that we noticed where MIR1 has been shown to be lowly expressed in the spinal fluid of patients with a variety of neurodegenerative diseases. Now, whether that's a cause or a consequence of the disorder, we didn't know. But we decided to undertake some research to dissect the function of MIR1 in this regard. So we used this polyglutamine YFP reporter, and it mimics Huntington's disease, where you have an abnormal expansion of glutamine repeats. And this disturbs the folding of the native protein that forms oligomers and aggregates, and impairs cellular function and survival. And Richard Morimoto from Northwestern did some really nice work on this in the early 2000s, where he expressed different numbers of polyglutamine repeats, and he found that if you have 35 of them, you don't see any aggregates. But if you have 40, you start seeing some aggregates, but you do also have some of these diffuse soluble elements here. Obviously, the further you get along here, the more toxic they are and the more they aggregate. So we thought this would be quite a nice model, this Q40 here, to examine the role of MIR1 in protein aggregation. So MIR1 is actually the only C. elegans microRNA that's 100% conserved through to mammals. Here are the sequences here, and this seed sequence will become important later. This is very important for targeting through from UTR of its target genes. So we crossed two MIR1 mutants into this Q40 strain, which has around 30 aggregates per worm, roughly. And in a MIR1 mutant, we observed 60 in these two different alleles here. We wanted to check that it wasn't just due to some deficiency in... Oh, sorry, one thing I haven't told you here is that these reporters are expressed in muscle, okay? We wanted to check that there wasn't just a defect in general microRNA expression levels in muscle that caused this phenotype. So we looked at this MIR80, which is a specific microRNA expressed in muscle, and we don't see any phenotype there. So previous work from Josh Kaplan's lab showed that MIR1 is expressed in two tissues, body wall muscle and pharynx. So we wanted to ask where MIR1 is acting to control Q40 or polyglutamine aggregation. So this is wild type. All of these are MIR1 mutants. This is the MIR1 mutant here without any transgenic rescue. We can rescue when you express MIR1 in muscle. We'll skip this one for a moment. You can't rescue if you express in pharynx or in an unrelated tissue, the intestine. We mutated MIR1 using these two residues here, such that MIR1 would be unable to interact with 3' UTR target genes. And we found that when we expressed this mutated MIR1, we were unable to rescue in muscle, as we did with the wild type. Okay? So MIR1 is important for regulating the aggregation of polyglutamine YFP. We wanted to ask how broad its effect may be. So we looked at a Parkinson's disease model. This is where alpha-synuclein is expressed in body wall muscle of worms. And this is where we're actually checking a behavior. So alpha-synuclein aggregates and causes defects in body wall muscle over time. And we can measure the effect of that looking at body bends of these animals. And we can see that both MIR1 mutant alleles here have a reduced body bend capacity compared to wild type when expressing alpha-synuclein, suggesting that they have more aggregates and it's toxic to these worms. Then we went more broad and we asked, okay, what happens in heat stress? So heat stress puts a huge amount of stress on the proteome and causes protein folding defects that need to be managed by various pathways. And we found that MIR1 mutants here are highly sensitive to heat stress. And we're basically looking at survival of animals after a particular temperature and time under heat stress. This phenotype here was fully rescued when you express MIR1 in body wall muscle, but not rescued when you express the mutated version of MIR1. So it seems that MIR1 is really broadly regulating protein aggregation in a variety of different models, as I've shown here. Now comes the tricky bit. So we know that MIR1 is regulating a target or targets to control proteotoxic stress. How do we identify targets? It's a very difficult proposition in microRNA research, and this slide took 4 years. So I'll take a little bit of time on it. So what would a target look like? Well, we'd expect it to be upregulated in a MIR1 mutant because microRNAs normally downregulate target genes. We'd expect knockdown of the target to suppress a MIR1 mutant phenotype. And we might expect that overexpression of a target phenocopies a MIR1 mutant. So we took a number of approaches, but I'm just going to talk about two here. First, we use an RNAi screen to suppress the MIR1 heat stress sensitivity phenotype. And we performed RNA sequencing of MIR1 and wild type animals to ask which genes are regulated by a MIR1 mutant. I'm not going to go into any of the data here. I'm just going to show you what we found. We found one gene that was very important. So this gene is called TBC7. We showed that TBC7 is upregulated with RNA sequencing, and we confirmed that by qPCR using two independent reference genes here. We see upregulation of TBC7 in the MIR1 mutant. Next, we showed that knockdown of TBC7 completely suppresses the MIR1 mutant heat stress phenotype. So in black are wild type animals with a control RNAi knockdown and TBC7 RNAi knockdown, no change. In a MIR1 mutant, which is sensitive to heat stress, we see around 50% lethality. This is completely suppressed when you knock down TBC7. Then we looked at polyglutamine aggregation by overexpressing TBC7, and we found that phenocopies of the MIR1 mutant phenotypes. It's a little bit complicated, but in black are wild type or Q40YFP animals. Here, overexpression of TBC7, line 1 and line 2. orange is MIR1 mutant Q40. We see aggregates here in the MIR1 mutant. Doesn't get exacerbated by overexpression of TPC7, suggesting at least that they may be in the same pathway, and with the other data quite solidified. So TPC7 is a bit of a mouthful. It's a RAB-GTPase activating protein of this particular family. I'm going to talk a little bit more about what this means in the slide after next. I just want to show you how we showed that TPC7 is a direct target of MIR1. So the sequence on the top here is the MIR1 sequence, showing the seed sequence in blue. We made a number of constructs with the wild-type TPC7 3' UTR in green, and then a mutated version here showing these two red residues that we mutated. So with the MYO3 promoter, which is body wall muscle specific, we express GFP with a UTR that doesn't have any MIR1 binding sites. We also made another construct where we had a MYO3 promoter driving mCherry, either with the TPC7 wild-type 3' UTR or the mutated 3' UTR. Then we express these in combinations in C. elegans and ask what happens. So on the top row here, we have GFP expression regulated by the UNC54 3' UTR. We can see that. We have cherry expression regulated by the TPC7 3' UTR wild type. We do detect some expression, but it's very weak. If we take this same line and place it in a MIR1 mutant background, we see de-repression of the mCherry, suggesting that MIR1 normally downregulates this 3' UTR. To confirm that, we took our mutated TPC7 3' UTR and drove it in wild-type animals, which has MIR1, but is unable to downregulate it because the MIR1 binding site has been mutated, and we see blazing expression there. So this together suggests that MIR1 is directly regulating the TPC7 3' UTR. Okay. So what is TPC7? Well, as I said, it's a RAP-GTPase activating protein. So RAP-GTPases cycle between either a GDP-bound off state or a GTP-bound on state. There are positive regulators called GEFs and negative regulators called GAPs. So TPC7 is a negative regulator of the RAP-GTPase, and it's switching this RAP-GTPase that is on the membrane and GTP-bound into a cytoplasmic GDP-bound version. RAB-GTPases are very well-known regulators of vesicle transport and fusion. And previous work, there are many different types of TPC proteins, but previous work has shown that mammalian TPC proteins are known regulators of autophagy. And in fact, they regulate autophagosome maturation and trafficking. So I want to just give you a very brief overview of autophagy, very little detail, but we know that it's a cytoprotective response to stress and requires these RAB-GTPases for vesicle trafficking and fusion. We start with this isolation membrane here, which elongates and starts engulfing cytoplasm organelles and protein aggregates until you form this autophagosome. This autophagosome then fuses with a lysosome to form an autolysosome where the contents are degraded and recycled. Okay? So RAB-GTPases are known to regulate the trafficking of these organelles and fusion between autophagosomes and lysosomes. So we wanted to ask whether TPC7 may have a role in autophagy, and that's why we see defects in clearance of these toxic proteins. I'm not going to go into details here again because I want to move on to the next stage, but what we found here was that in heat stress, MIR1 mutants have a reduced what we're calling autophagy index, so they're poorer at mounting an autophagy response to heat stress. But interestingly, they don't seem to have any phenotype in normal conditions, control conditions at 20 degrees. And we found exactly the same phenotype when you overexpress TPC7 in body wall muscle, which is the tissue in which MIR1 is regulating TPC7 expression. Okay, so now we have this pathway where MIR1 downregulates the expression of TPC7 to control autophagy and mediate prototoxic stress responses. Could this mechanism be conserved? So we looked at target prediction programs in Drosophila and humans, and the Drosophila homolog of TPC7 is called Skywalker, and this is a predicted target of MIR1 in Drosophila. In addition, the human version, a human homolog of TPC7 is called TPC1D15, which is also a predicted target of MIR1 in humans. So now, in collaboration with David Rubenstein's lab, we have analyzed this potential regulatory relationship in mammalian cells. So the first experiment we did was to take HeLa cells and overexpress MIR1 in these cells and ask whether we changed TPC1D15 expression. Luckily, there was an antibody available for this, and we can see that overexpression of MIR1 can downregulate expression of TPC1D15. Next, we performed very similar to experiments in the worm using sensor constructs, where we have the human MIR1, TPC1D15 wild type and mutated 3' UTRs. We found that overexpression of MIR1 in HeLa cells can downregulate the 3' UTR of TPC7, but it cannot downregulate the 3' UTR of mutated TPC1D15. Okay, so we're seeing very similar theme to C. elegans. Next, we wanted to ask whether MIR1 regulation of TPC1D15 is important for autophagy. We did many experiments here, but I want to show you this traffic light experiment, which makes it very easy to comprehend. What this assay here is, we have LC3, which is a very important autophagy protein, that's fused to GFP and M-cherry. In autophagosomes, both GFP and M-cherry are fluorescent, and they appear yellow. When these autophagosomes fuse with a lysosome, the environment within the autolysosome is acidic, and that quenches GFP, so these autolysosomes appear red, and then they go on to degradation. So in normal basal conditions, you normally see this kind of phenotype here, where we see more red lysosomes, around 2 times the number of autolysosomes to autophagosomes. That's a standard picture that we observe. So we use this assay to first ask what effect does MIR1 have on autophagy flux, and we see an increase in the number of autolysosomes, i.e. more red vesicles, more autolysosomes here, when you overexpress MIR1, meaning that MIR1 is processing autophagy more efficiently. When we did the reverse experiment and overexpressed TPC1D15, which is what you'd be expecting with the MIR1 knockout, we saw a very dramatic phenotype. So what we see here are these huge yellow autophagosomes that are not going anywhere. They're not fusing with the lysosome to form autolysosomes here. And we hypothesize this is why we see problems with aggregation. So taken together, these data suggest that MIR1 is an efficient driver of autophagy flux, and that TPC7D115 overexpression blocks autophagy. So now we're going back and forth between worm and mammalian work. So our previous work in worms showed that MIR1 regulation of TPC7 is important for regulation of polyglutamine YFP in C. elegans muscle. So we wanted to ask whether Huntington aggregation in mammalian cells is also regulated by this pathway. So we showed that again in HeLa cells, where if you overexpress MIR1, these 2 different independent constructs, we see a reduction in Huntington aggregation in these HeLa cells. I won't show you the data, but this reduction is absolutely dependent on autophagy. We also downregulated by siRNA the TPC1D15 expression, and this has exactly the same phenotype, which is what you'd expect by overexpressing MIR1 and knocking down TPC, would see the same effect. Importantly, we showed that when we overexpress TBC1D15, it prevents the ability of MIR1 to reduce Huntington aggregation. So this is empty vector control, where we see overexpression of MIR1 reduces Huntington aggregates. This is cells that are overexpressing TBC1D15, and MIR1 is unable to have any effect on this because this construct doesn't have a MIR1 binding site. So finally, we have this pathway where MIR1 is regulating this protein here to control autophagy and reduce Huntington aggregates. We wanted to go one step further and ask whether we could possibly use this regulatory relationship and modify it as a therapeutic. Now, there are a couple of stories that came out over the last few years that suggest that interferon may be used for this purpose. So this is a cytokine that a previous paper had shown in liver cells that MIR1 is regulated by interferon. In addition, our collaborators previously showed that lack of interferon causes neurodegeneration and dementia. So we want to examine this in mouse cortical neurons. So what I'm showing you here is application of interferon and the expression of MIR1. And we can see that it is responsive to interferon. Similarly, when we look at TBC7 protein levels, this is decreasing over time with interferon expression, presumably through upregulation of MIR1. We also showed that expression of interferon beta reduces Huntington aggregates. And importantly, this is dependent on MIR1. These black bars here are a reduction of Huntington aggregates by interferon. And the interferon effect on Huntington aggregates is reduced when you knock down the expression of MIR1 in these cells, suggesting that interferon is upregulating MIR1 here to control removal of Huntington aggregates. So, taken together, we've identified this pathway where interferon controls the expression of MIR1, downregulates the expression of this important controller of autophagy, which is able to remove, or at least reduce, aggregate formation for Parkinson's disease, Huntington's disease, and potentially other protein aggregation disorders. So I think I'll leave it there. Just like to acknowledge a great group of people in the lab, quite PhD student heavy at the moment, but that's okay, keeps me busy. I'd like to thank initial funding from the European Research Council in Europe, and then the National Health and Medical Research Council in Australia, and also some very nice local funding from the Victorian government that I received when I first started my lab. I'd very much like to thank our collaborators, David Rubenstein and Patrick Eilesko, who have really pushed this, the second project forward immensely. And I'd like to thank you for your attention.
The first story
Yes, if I can remember.
is the insulin signaling activity of the BAG neuron related to its oxygen and carbon dioxide sensing activity?
Right, good question. Absolute. Well, I'd like to say no. I think no. So we've tested a GCY9 mutant that's unable to sense CO2, and GCY3133 that's unable to sense oxygen in the BAGs, and they have no expiration phenotype at all. So we haven't looked at whether dysregulation of those receptors control insulin expression actually. Haven't looked at that, but the actual expiration phenotype, there's no similarity here. So we think there's a separate function. Yeah. Yeah.
So
Hi.
I have two extremely tenuously related questions.
Okay.
They somehow in my mind relate, but I'll ask them both
Great.
in case they connect. So since Max asked you about the first, I'll ask you about the second.
Mhm.
More in my memory. So before you showed us the evidence that MIR1 has TBD1 or whatever as a direct target,
Yep.
I was wondering
set out as your screen to look for things that are increased in expression, to look for targets, if I remember the strategy.
Yep.
So what about the idea that unrecognized targets lead to cellular change, pathology, etc., and then things come up secondarily,
Mm-hmm.
right?
Mm-hmm.
So then, if I recall, your screen was by qPCR.
The screen was by RNA sequencing, but that's
RNA
fine.
sequencing
Yep,
at
yep.
RNA level, which is the real point.
Yep.
And if I don't totally misunderstand,
Mm-hmm.
mirrors frequently happen at the next step of regulation of translation from.
Yes.
So I'm wondering, might there be still the possibility that there are critically regulated pathways that show you this phenotype without the direct regulations that you would have found by your RNA-seq
Yeah.
screen?
Yeah.
And maybe the second half of the question, in case they connect,
Mhm.
is I'm wondering about the speculations about neurodegenerative disease and the idea that there are these mutations in every cell in the body and every neuron of the, I think, many thousands of distinct kinds of neurons, and only a relatively narrow set of cortical neurons and a set of striatal neurons are the, let's say, centrally
Mhm.
aggregating and abnormal. How does the HeLa cell set of experiments weigh into that?
Yeah.
And maybe that gets to what could be some of those things in the first long half of my question that are regulated, that get to cell specificity, really.
Right.
Does that make sense?
I think I need to go and have a sit down. No, it's, I get it.
Let's
No, so I think the first part of the question, we do know that microRNAs can control RNA stability, which is why we may see more of the TBC7 in the RNA sequencing. That's why we look for that. Obviously, yeah, it would have been nice if we could look at the proteome in that context. But we kind of, to be brutally honest, we did the RNAi screen first and identified a bunch of, identified 4 factors that suppress the MIR1 mutant heat stress phenotype when you knock them down. Then we did the RNA sequencing, and only TBC7 was regulated. Doesn't mean that those other guys are not doing anything, but they could be in the pathway, they could be in a separate pathway, right? That you improve another pathway and you remove the aggregates and you promote survival. So that's definitely a possibility. I think, always with these cases, you have a simple pathway, but it's never as simple as that, right? And we all know that, but we try and explain what we have in the easiest way possible. Whether that's the best way, I don't know, but we have this direct relationship, and we see that they're both. I think this is why we knock down and overexpress in multiple different ways with controls in different cells to enhance our faith in the data. So your second question was on the.
Well, the aggregation, the cell type specificity, the insights brought by HeLa versus specific populations of neurons, and broadened now by your answer to the first part, is can one then intuit that the interferon
Right.
is acting through this
Right.
pathway versus all these hidden ones
Right.
that we may not have thought about yet?
Right. So the interferon we've shown functions whether you look at HeLa cells or neurons.
Right.
So it has the same effect on MIR1 expression and on TBC1 D15 expression in both those very different cell types. So that gives us some confidence. May not be regulating the same pathway in other neurons or cells. We don't know. I think we need to be a bit careful with this pathway because MIR1 overexpression can cause cancer. So we need to be careful about how we think about potentially using this pathway as a therapeutic. And yeah, as with any therapy, I suppose you don't know how manipulating the pathway will affect different cell types. So if you can try and manipulate locally, that may be advantageous. But yeah, I don't know whether I've answered your question at all. But
Yeah, I don't want to take time from
That's-
I have a
we can-
secondary follow-up on that, but there's
Hi.
Thank you. I think both stories are very interesting to me.
Thank you, I do too.
One question for the first one, because you actually, in the first story, you are talking about the brain and gut communication, and I think for how the brain controls the intestine, I think that part is very clear. And I think you have used one way to actually use a very rich food to feed the
Yep.
enzyme to see how the lipid increase in the intestine and how the PET1 and INS1 actually reduced in the brain.
Yep.
And how do you know that? How do you exclude the possibility that actually the food actually just directly increase the gene expression in the brain? It's not by actually go to the intestine and then feedback to control.
Absolutely. So we're doing those experiments right now. So we're using other manipulations to reduce the fat levels in those conditions to see whether we can reverse the expression. So we're definitely doing that right now.
Actually, yeah, it's similar to this, related to this question.
Yeah.
Did you try to starve the ETS5 mutants, and then that would usually reduce the fat content to see that if that rescue behavior?
Yeah.
Because it seems there is this feedback loop, right?
Yep.
So ETS5 regulate fat, and then we know that fat may regulate to exploration. But then the exploration will regulate to fat again, right? So
Yeah, sure. Yeah, we did a quite acute experiment for the paper where we used this drug called Astrianam. So if you place bacteria on Astrianam, they don't divide, and they form filamentous bacteria. So the worm can smell the food, we think, as normal food, but it can't eat it. If you put ETS5 mutants on that bacteria, they immediately start exploring. So their behavior is wild type. And I kind of see that they're anticipating that they're going to be starving because they can't eat this food, and they start exploring. So even though they've got high fat at that point, they switch their phenotype very quickly because they sense that they're not eating.
Right, so even before the fat
Is
the
depleted,
fat content,
yeah.
got it.
It can deplete quite quickly within an hour or so, but I think they're sensing, okay, these conditions are not optimal long term, and I need to explore.
So it's very the sensory response.
Yeah.
Related,
And we don't understand biology regarding that.
Another thing is that you can rescue the INS1 by expressing it back. Did you try to see the non-rescue the INS1 in other neurons?
That's right. So we did that. Yeah. So we expressed in either multiple neurons or even just a couple of different pairs of neurons, and we could not rescue whatsoever. So it seems that flooding worms with INS1 isn't sufficient. So we've shown that if you overexpress INS1 in the BAG neurons at a very high level, they rest at the L4 stage.
Right.
Okay. More recently, when we're looking at DAF2, if we overexpress DAF2 in AIB, they arrest at the L4 stage. So we're potentially thinking the level of expression of these two genes in these two different cell types are regulating. So it's, yeah.
I think one common comment that we also got from our study is that neuropeptides doesn't have to work synaptically.
No.
Right, and then so then what gives this cell type specific function of the neuron that secreted one peptide control one behavior versus other?
That's right. That's right. I have a feeling it's one's acting locally, but we need to address that. Not easy. Yeah, because you can't tag it.
Right, yeah, exactly.
Right.
Just one
Max.
more on the
Yeah,
second
yeah.
half of the talk.
Yeah, cool.
So I may have just misunderstood.
Yep.
Can you say a bit more about how you think TBC7 is acting in autophagy? I was confused because
Yeah.
it seemed like you were saying it promotes autophagy, but its overexpression prevents autophagy.
Right.
it's
So
positively and negatively controlling autophagy?
if we look at here, so it's a negative regulator of Rab GTPases. So if you have too much TBC7, which is what you'd have in a MIR1 mutant, you're switching this Rab GTPase to off, which means that the fusion of these, the autophagosome and lysosome is not occurring as efficiently, and they're stalling at the autophagosome stage. We haven't shown that, but we do know that Rab7, which is a Rab GTPase target of GAP, if you knock that down, they're very similar phenotypes to when we overexpress TBC7.
Okay, yeah, all right, thank you, Roger.
Thank you.